Hello everyone, welcome to your own YouTube channel Shantanu Bioprep Life Science. I hope you are doing great. Today we are going to discuss Krebs cycle in detail. Okay, so I hope previous videos were very helpful for you, especially for GATE Life Science biochemistry students, those who are taking biochemistry subject in GATE Life Science. And biochemistry also has good weightage in all life science examinations. Okay, so starting with the Krebs cycle today. Starting with the salient features of Krebs cycle or characteristics of Krebs cycle. Krebs cycle is also known as TCA cycle or citric acid cycle. Why it is known as TCA cycle? Because Krebs cycle will have some intermediates which will have 3 carboxyl groups. Which are those intermediates? Citrate, cis-aconitate and isocitrate, which we are going to see in the upcoming slides. Okay, that's why it is known as TCA cycle, because the intermediates, few of the Krebs cycle intermediates will have 3 carboxyl groups. Okay, location is in mitochondrial matrix in the case of eukaryotes, because we know that in eukaryotes membrane bound cell organelles are present, whereas in prokaryotes membrane bound cell organelles are not there. That's why it will take in cytosol. 65 to 70% ATP is synthesized in Krebs cycle, which accounts for 2/3 of energy which will be produced through Krebs cycle. Okay, so 65 to 70. From where we are getting this number? We have already seen that in glycolysis 8 ATPs are produced. Via pyruvate oxidation 6 ATPs will be formed. And we know that one molecule of glucose when oxidized will generate 38 ATPs, which we are already studying it from 11th or 12th standard. So 38 minus 14. 8 from glycolysis, 6 ATPs from pyruvate oxidation. 8 plus 6, 14. 24 ATPs will be formed from Krebs cycle. So can we say that if we want to calculate how much, or we can say that 24 is what percent of means multiplication sign, 24 ATPs is what percent of 38 ATPs. So if you calculate this, you will get answer as approximately 66%. Okay, so this number is coming from here, 66%. I have discussed it and solved it for you. If a question is asked in the exam, glycolysis will account for what percent of energy when one molecule of glucose will get oxidized. So how will you calculate? We know that in glycolysis net ATP gain is 8. So can we say that 8 is what percent of 38? So whatever answer you will get for X, you will get directly the percentage. Okay, X percent of ATP will be synthesized in glycolysis. This is how you can calculate it very easily. Okay, 2/3 of total oxygen in the body is consumed by Krebs cycle, and Krebs cycle occurs only in the presence of oxygen, although there is no direct involvement of oxygen in Krebs cycle, unlike in electron transport chain. You should remember that this is very important point. You should remember that although 2/3 of total energy is consumed in Krebs cycle, but oxygen is not directly participating in Krebs cycle, or oxygen is not directly involved in Krebs cycle. See, in Krebs cycle, NADH, FADH2 will be produced, which will pass through electron transport chain, and then it will result in the formation of ATP. In the electron transport chain, we have studied it that the last electron acceptor in electron transport chain, or terminal electron acceptor in electron transport chain, is oxygen. So if Krebs cycle is initiated and if NADH and FADH2 are formed, it is going to enter electron transport chain. I'm going to synthesize ATP, but for that oxygen will also be utilized. Then only the electron transport chain can be completed for ATP synthesis. So indirectly, Krebs cycle is also using oxygen, but it is not directly using oxygen. Okay, electron transport chain is directly utilizing oxygen because the last electron acceptor is oxygen. Okay. Next, oxaloacetate plus acetyl coenzyme A will be converted into citrate, which will be catalyzed by an enzyme citrate synthase. This step is condensation. This is 4 carbon compound. This is 2 carbon compound. This is 6 carbon compound. So condensation is going to occur and it will result in the formation of 6 carbon compound molecule, that is citrate. In the citrate, you can observe that there are 3 carboxyl groups. That's why the name of Krebs cycle is also TCA cycle, tricarboxylic acid cycle. Okay. This is the structure of oxaloacetate, which you need to remember because there is no shortcut. Okay. Acetyl coenzyme A structure is this. There will be liberation of COASH. Okay. So if we draw the structure of acetyl coenzyme A over here, so if I draw CH3 like this, and this rest of the structure, one hydrogen is getting lost because, as you can observe it from here, so this hydrogen will be, suppose this hydrogen is lost, and along with it this is also lost. What is left? CH2CO, that is this group. This group will combine with oxaloacetate and form this. Okay, very easy. Only we have to add this structure with this structure. We will get citrate. Okay, easy. Next, citrate will be converted into cis aconitate. This is dehydration reaction because there is a removal of water molecule. There is a removal of water molecule. Enzyme catalyzing this is aconitase. Citrate, which we have discussed, how is it formed in the last slide. There will be removal of water molecule. So if OH will be removed from here, hydrogen atom will be removed from here, whatever the structure is left, if we write it, it will be of cis aconitate. Why double bond is present? Because we have been discussing this from the previous lectures that carbon atom has to form 4 bonds. So if we add double bond over here, these 2 bonds will be there. This will be 3rd bond and this will be 4th bond for this carbon atom. Okay, for this carbon atom it will be also balanced. This is 1, 2, and these 2 will be 4 bonds total for this carbon atom. Okay, if there was no double bond, then if there was this single bond instead of double bond, then the case would have been this carbon atom would have formed only 3 bonds, which is not correct because carbon atom has to form 4 bonds. That's why there is double bond over here. Okay, cis aconitate will be converted into isocitrate by an enzyme aconitase, and here there will be addition of water molecule. That's why the name is or that's why the name of this step is hydration. Cis aconitate. Okay, we will add water molecule in it. So H is here, OH is here, water molecule have been added, due to which the structure will now change to this. This is isocitrate, which is an isomer of citrate. If you individually count the number of carbon, oxygen and hydrogen, you will get the same for these two. So the chemical formula of citrate and isocitrate will be same, but their structures or the number of or the arrangement of atoms will be different in their structure. So the structures will be different, but chemical formula will be same for citrate and isocitrate. That's why they are isomers of each other. Okay. Isocitrate will be converted into oxalosuccinate, which will be catalyzed by an enzyme isocitrate dehydrogenase. And here NAD plus is a coenzyme of isocitrate dehydrogenase. It is getting converted into its reduced state NADH plus H plus. So this is a structure of isocitrate. One hydrogen atom is going to be removed. So there is oxidation of isocitrate. So if a new student is watching this lecture, oxidation is removal of hydrogen atom and electron. Reduction is acceptance of hydrogen atom and electron. Okay. And if you are new student, please do watch the video lectures right from the beginning so that you can understand the concept very easily. Okay. So there is removal of this hydrogen atom, which means oxidation of isocitrate is going to occur. This hydrogen atom will be accepted by oxidized NAD plus, and it will get converted into its reduced form, that is NADH plus H plus. Why reduced? Because it is accepting hydrogen atom. And we have discussed the acceptance of hydrogen atom and electron is known as reduction, whereas loss of hydrogen atom and electron is known as oxidation. Okay, so this is oxidized state, this is reduced state. After acceptance, after this hydrogen atom is lost, we will get the structure like this. Okay. Oxalosuccinate will be converted into alpha ketoglutarate by an enzyme isocitrate dehydrogenase. In this reaction, there will be loss of CO2 molecule, that is decarboxylation is going to occur. So from the molecule, one carbon will be less. For example, oxalosuccinate is of 1, 2, 3, 4, 5, 6 carbon molecule. So if CO2 is getting removed, number of carbon which will be left in the molecule should be 5. 6 minus 1, 5. As we can count it, 1, 2, 3, 4, 5th carbon. Okay. So oxaloacetate will be converted into alpha ketoglutarate after removal of CO2. So if CO2 will be liberated, this oxalosuccinate structure will be converted into alpha ketoglutarate. Okay. Now you must be wondering, here it was CH and here it is CH2. Why? Because carbon has to form 4 bonds. Simple explanation. Okay. Alpha ketoglutarate will be converted into succinyl coenzyme A, which will be catalyzed by an enzyme alpha ketoglutarate dehydrogenase. It is oxidative decarboxylation step. Okay. Why? We will understand it. From this alpha ketoglutarate, there is a removal of CO2 molecule, due to which the name will be decarboxylation. CO2 is getting removed. There is also an addition of coenzyme A. In coenzyme A, this hydrogen atom, this one shown in the circle, will be accepted by oxidized NAD plus and it will get converted into its reduced form, that is NADH plus H plus. And after the removal of CO2 from alpha ketoglutarate, this structure is nothing but succinyl coenzyme A. This coenzyme A, that is CoAs, will be attached over here, and what we will get is succinyl coenzyme A. Memorizing the structures of Krebs cycle is little difficult because we will not get any hint from the names. Because in Krebs, in glycolysis, the names were like glucose 6-phosphate. So we know that at 6th position there will be phosphate group. But here the names are like citrate, alpha ketoglutarate, fumarase, malate. So you will not get any hint from here, from the names, from their names. Okay, so you need to remember it if you want. Okay, very rarely the questions are asked on structures. You can skip structures if you want. Okay, but the concept should be clear. So now you know that how succinyl coenzyme A is formed. In the next step, succinyl coenzyme A will be converted into succinate by an enzyme succinate thiokinase or succinyl coenzyme A synthetase. Coenzyme A group is getting removed. GDP is getting converted into GTP, that is phosphorylation is going to occur. This is very important point. It is the only substrate level phosphorylation step in entire Krebs cycle. So you may get a question from this point. So we will understand the phosphorylation step here in detail. Here GTP is formed, but this GTP can be again converted into ATP. This will be catalyzed by an enzyme nucleoside diphosphate kinase. What it will do, it will take a phosphate group from GTP and it will add a phosphate group on ADP. If a phosphate group from GTP is removed, we will get GDP. If a phosphate group is added to ADP, we will get ATP. And this is a reversible reaction. That's why I have shown two-headed arrow over here. If we want to convert ATP into GTP, it is also possible. This enzyme can remove phosphate group from ATP and add it to GDP. If a phosphate group from ATP is removed, it will be converted into ADP. If a phosphate group is accepted by GDP, it will get converted into GTP. So this is a reversible step. Okay. So as we can see that from the succinyl coenzyme A is getting released. So there will be removal of this group and GDP is getting converted into GTP. Okay. So after the removal of this group, we will get the structure of succinate. Structure of succinate is very much easy to remember. First and last carbon will have carboxyl group, and second and third will have CH2 group. Okay. Next, succinate will be converted into fumarate, which will be catalyzed by an enzyme succinate dehydrogenase. The step name is oxidation. So we can get an idea from here that oxidation means there will be removal of hydrogen atom, and there will be someone who will accept those hydrogen atom and it will get converted from oxidized state to reduced state, which we have been discussing it from the previous lectures. So FAD is present in oxidized state. After acceptance of hydrogen, it will get converted into reduced state, that is FADH2. Succinate structure, we have discussed it in the last slide. So if I draw structure of succinate like this, if hydrogen atoms, 2 hydrogen atoms are getting removed, these 2 hydrogen atoms will get accepted by FAD and it will get reduced and form FADH2 upon the acceptance of 2 hydrogen atoms. and we will get structure like this after the removal of hydrogen atoms. Okay, why double bond? Because carbon atom has to form 4 bonds. So you can count it and observe. In this step, fumarate will be converted into malate, which will be catalyzed by an enzyme fumarase. In this step, hydration or addition of water molecule will occur. Okay. So fumarate molecule is this. If we add water molecule, here OH group and here H group, we will get malate. Very easy. Okay. In malate will get, in the last step, that is malate will get converted into oxaloacetate, which will be catalyzed by an enzyme malate dehydrogenase. This is also an oxidation step. Okay. It is also an oxidation step. In this, malate or malate dehydrogenase enzyme will oxidize the malate. This hydrogen atom will be removed and it will get accepted by NAD plus, and then it will get converted into, or it will get reduced into NADH plus H plus. After the removal of hydrogen atom from malate, the same structure or the structure left is nothing but of oxaloacetate. So we are getting oxaloacetate. The first substrate was also, first reactant was also oxaloacetate, and the last product is also oxaloacetate, because this is cyclic type of metabolic pathway which we have discussed in detail in first lecture, that is introduction of metabolism. If you haven't watched that video, please make sure to watch it so that you will get to know all the concepts very clearly. Okay. So if we quickly summarize how many NADH, how many FADH2, how many GTPs are getting formed, we can observe that so this is first NADH, this is second NADH, this is one GTP, this is one FADH2, and this is third NADH. So total NADH are, total 3 NADH molecules are getting formed, one GTP and one FADH2. These molecules are getting formed from one molecule of acetyl coenzyme A. Okay, from one molecule. We know that when NADH passes through electron transport chain, it will form 3 ATPs. So after 3 NADH passes through electron transport chain, we will get 9 ATP. 1 GTP will result in 1 ATP. And 1 FADH2, when passes through electron transport chain, it will result in the formation of 2 ATP. Total 12 ATPs from one molecule of acetyl coenzyme A. And we know that 2 molecules of pyruvate are formed in glycolysis. 2 molecules of pyruvate will be oxidized into acetyl coenzyme A in pyruvate oxidation. 2 molecules of acetyl coenzyme A will enter Krebs cycle. Okay, from one molecule of acetyl coenzyme A, we are getting 12 ATPs. From 2 molecules of acetyl coenzyme A, we will get 24 ATPs. Okay. Inhibitors of Krebs cycle. Fluoroacetate is a pesticide which inhibit aconitase enzyme. Aconitase is non-heme ion protein. This was asked in CSR net technique question part. It was experimental based conceptual question in part C. Okay, so this you have to remember that aconitase is non-heme ion protein. Fluoroacetate, which is a pesticide, it will inhibit aconitase by conversion into fluoroacetate. Fluoroacetate will get activated into fluorocitrate and it will behave as a potent inhibitor of aconitase enzyme, due to which Krebs cycle will get inhibited. Malonate is a competitive inhibitor and it will inhibit succinate dehydrogenase. Succinate dehydrogenase. So malonate will be competitive inhibitor of succinate. We have discussed competitive inhibitor also previously. Let me quickly summarize it for you. So if this is an enzyme succinate dehydrogenase with its active site, and if we draw the substrate, suppose this is succinate, competitive inhibitor will always, or it will mostly resemble its structure will resemble with substrate. Okay, so the inhibitor will look exactly like the normal substrate. This is inhibitor. Now what will happen if they are looking similar or identical? They both can bind with the active site of an enzyme. So here enzyme is succinate dehydrogenase, normal substrate is succinate, inhibitor is malonate. Inhibitor is malonate, which is exactly looking like the normal substrate, that is succinate. So if they are both looking identical, they can compete with each other to bind with the active site. Succinate will say I will bind, malonate will say I will bind with succinate dehydrogenase enzyme. So there will be competition among them. Okay, that's why we are calling it as competitive inhibitor. Malonate is a competitive inhibitor of succinate, and it will inhibit succinate dehydrogenase enzyme. Arsenite is non-competitive inhibitor, and it will inhibit alpha ketoglutarate enzyme. Okay, so this much only you have to remember in inhibitors of Krebs cycle. Role of vitamins in Krebs cycle: thiamine, it is acting as thiamine pyrophosphate, which we have discussed it in pyruvate oxidation. What is TPP? Thiamine is vitamin B1, and it is coenzyme for alpha ketoglutarate dehydrogenase. Riboflavin is vitamin B2. It is acting, FAD is a coenzyme for succinate dehydrogenase. So if FAD is less, sorry, if vitamin B2 levels are less, if vitamin B2 levels are less, FAD amount will also decrease. Okay, so there will be less FADH2. Due to this, there will be less ATPs formed. That's why riboflavin should be in sufficient amount. Vitamin B2 should be in sufficient amount. Otherwise, the amount of ATPs will be reduced. Okay. Niacin is vitamin B3. Okay. And NAD plus from vitamin B3, which is derived. This NAD plus is an electron transfer for isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. It is acting as a coenzyme or electron transfer. So if vitamin B3 levels are decreased, NAD plus levels will also decrease, due to which there will be less NADH formation, due to which amount of ATP will be drastically reduced as compared to FADH2. Why? FADH2 will only result in the formation of 2 ATPs, but NADH will result in the formation of 3 ATPs through electron transport chain. So much lowering of vitamin B3 levels is much dangerous as compared to vitamin B2 levels. Okay. Pantothenic acid or vitamin B5 acting as coenzyme A is attached to acetyl coenzyme A and succinyl coenzyme A. This is acetyl coenzyme. That is a spelling mistake. Okay. If acetyl coenzyme A is not produced, Krebs cycle will not occur. Why? Because we know that oxaloacetate will combine with acetyl coenzyme A and then citrate will be formed. If acetyl coenzyme A levels are very much low, it won't be able to bind oxaloacetate properly, or it will not occur at all. Okay, due to which Krebs cycle will not occur. If Krebs cycle will not occur, there will be much less formation of ATPs, because we know that through Krebs cycle 24 ATPs are formed. That is 65 to 70% of energy is formed through Krebs cycle. Okay. Now we will discuss few important points. Krebs cycle is the most important central metabolic pathway connecting mostly all individual metabolic pathways directly or indirectly. Krebs cycle is amphibolic in nature. It can serve as both anabolic as well as catabolic metabolic pathway. In the first video, that is introduction to metabolism, we have discussed it in detail that those metabolic pathways which can be of either anabolic or catabolic type, or which can play both anabolism or metabolic, which can play both anabolism as well as catabolism, such pathways are known as amphibolic pathways. Okay, here amphibolic. Since Krebs cycle has anabolic nature, the intermediates later will get depleted. Such reaction where these intermediates will get replenished or formed again are referred to as anaplerotic reactions or anaplerosis. This point we will discuss in next slide. Okay. There are three steps or three reactions which we need to study. Pyruvate will be converted into oxaloacetate, which will be catalyzed by an enzyme pyruvate carboxylase. In this, there will be addition of carbon dioxide. So, addition of carbon dioxide means carboxylation. Okay, and there will be utilization of 1 ATP to form oxaloacetate. So, oxaloacetate is getting replenished by this reaction. So, these three reactions are not much important, you can skip it. Okay. This step occurs in animals. Next reaction is that phosphoenol pyruvate will be converted into oxaloacetate by an enzyme PEP carboxylase. This will occur in plants and bacteria. So in the case of animals, oxaloacetate will be produced from pyruvate by this reaction. In plants and bacteria, oxaloacetate will be produced from phosphoenol pyruvate by this enzyme. Okay. PEP carboxylic kinase can also convert PEP into oxaloacetate. Okay. And this step, this reaction you can also remember if you want. Pyruvate will be converted into malate by an enzyme malate dehydrogenase. Here it will be addition of CO2 group or addition of carbon dioxide, and 1 NADPH will be used. Okay. Now we will understand anaplerotic reactions in detail. If we know that acetyl coenzyme A, oxaloacetate and acetyl coenzyme A will combine and result in the formation of citrate. So this is an intermediate of Krebs cycle. Citrate will be in turn used in the formation of or in the biosynthesis of fatty acid and sterol. If this intermediate of Krebs cycle is getting used in the biosynthesis of fatty acid and sterol, the levels of citrate will begin to decline after some time, because it is used in the biosynthesis of fatty acid or sterol. It is used in the formation of fatty acid and sterol. So the intermediate of Krebs cycle, that is citrate, its level will going to be decreased after some time. So such chemical reaction where a reactant will produce intermediate, that is citrate in this case, to replenish or to again increase its levels, is known as anaplerotic reaction. Okay, this A will be substrate or reactant. Citrate will be product of anaplerotic reaction. Okay. Similarly, in alpha ketoglutarate, it will get converted into glutamate by transamination reaction. Glutamate will be used in the synthesis of non-essential amino acid and purines. If this continue, there is obvious thing that the intermediate of Krebs cycle, that is alpha ketoglutarate, its level will going to be decreased. Such reaction which will result in the formation of alpha ketoglutarate, due to which the amount or levels of alpha ketoglutarate will again begin to rise. Such reactions are known as anaplerotic reactions. The main aim of this reaction is to replenish or to again form the intermediate so that it can reach or it can rise its level, is referred to as anaplerotic reaction. Okay. Succinyl coenzyme A will also be used in the formation of heme. Okay. Malate will be converted into pyruvate by an enzyme malate dehydrogenase, which we have seen, and it is reversible reaction. Similarly, oxaloacetate can be converted into pyruvate by pyruvate carboxylase, which we have seen over here. It is also a reversible step. Oxaloacetate will be converted into aspartate by transamination reaction. We are going to discuss transamination reaction when we will study amino acid metabolism. This aspartate which will be used in the formation of non-essential amino acid and pyrimidine. So this was the anaplerotic reactions. Anaplerotic reaction. Okay. So some content was missing behind my video, which was glutamate will be used in the formation of non-essential amino acid and purine. Okay. Next, regulation of Krebs cycle. There are 3 regulatory enzymes in Krebs cycle which will control and regulate the rate of Krebs cycle: citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. ATP, NADH, acetyl coenzyme A, and succinyl coenzyme A will inhibit citrate synthase. Now let us understand the logic behind it. Okay. If ATP levels in the body is in high amount or is in sufficient amount, should the Krebs cycle occur? Krebs cycle should occur or not? Krebs cycle should not occur. Why? In the Krebs cycle, 65 to 70% ATP is formed in Krebs cycle. So if ATP is already present in high amount or sufficient amount, there is no need of formation of further ATPs, because there will be wastage of ATPs. What the body will try, it will try to suppress the further ATP synthesis, because ATPs are present in sufficient amount right now. The body will try to prevent the use of fresh intermediates, and the body will say that there is no need of ATP synthesis right now, we will just prevent the Krebs cycle, and when there is actually a need of energy in the body, we will again start Krebs cycle. So if there is enough ATP, it will try to, or it will inhibit citrate synthesis so that the Krebs cycle will not occur. Similarly, NADH, we know that NAD plus, when it accepts hydrogen ion, hydrogen atom, it will get converted into NADH. This NADH will pass through electron transport chain and result in the formation of 3 ATPs. If the levels of NADH is in high amount, which means ATP synthesis is going to occur from this molecule. That also 3 ATPs from one molecule. So the ATP will be ultimately the ATPs will ultimately rise. So there is no need of formation of excess ATPs because NADH levels are already in sufficient amount. The body will try to inhibit the Krebs cycle to prevent the formation of excessive ATPs. Okay, and the body will try, or the body will only activate Krebs cycle when there is actually a need of energy inside the body. So NADH will also inhibit citrate synthesis. Similarly, acetyl coenzyme A. If the level of acetyl coenzyme A in the body is in high amount, the body will know that acetyl coenzyme A will pass through Krebs cycle. Then the NADH, FADH2, GTP will pass through electron transport chain and it will form ATPs. So if this is acetyl coenzyme A is in high amount, it will pass through Krebs cycle and result in the formation of further ATPs. So the body will try to inhibit the usage of this fresh acetyl coenzyme A, and the body will say that let the old acetyl coenzyme A to be used in the Krebs cycle, and when the body will actually need energy, it will use this fresh acetyl coenzyme A. But now, since the acetyl coenzyme A is present in high amount, it will stop the next Krebs cycle and it will only allow the fresh Krebs cycle to begin when there is actually a need of energy inside the body. So now acetyl coenzyme A will inhibit citrate synthesis. Okay. Similarly, succinyl coenzyme A, it will tell the body that I am present in high amount. Let me complete first. Okay. Then you start with fresh succinyl coenzyme A. Okay. So this is the logic behind it. Similarly, isocitrate dehydrogenase will be inhibited by ATP and NADH and it will get activated by ADP. Again, same logic, ATP concentration amount is high. The body will say that ATPs are already in high amount. Then why to stimulate the Krebs cycle in order to produce excess ATPs? There is no need right now because there will be wastage. So ATP and NADH will inhibit isocitrate dehydrogenase, due to which Krebs cycle will not occur. Okay, and there will be further, or the excessive ATP formation will be prevented. But the levels of ADP when rise, it will stimulate Krebs cycle by the activation of isocitrate dehydrogenase. Why? If the ADP levels are increased, we know that ADP plus inorganic phosphate will result in ATP formation. If ADP levels are increased and not ATP levels, this suggests that the body is in energy deficient condition and the body actually needs to synthesize ATP. So the body will try to activate the Krebs cycle so that 24 ATPs can be produced, and which is what we want right now because the body is in excessive deficient condition or excessive energy deficient condition because the ADP levels are already very much high. So it will try to activate isocitrate dehydrogenase. It will promote the activation of Krebs cycle, due to which ATP synthesis will occur. Okay. Alpha ketoglutarate dehydrogenase will be inhibited by succinyl coenzyme A and NADH. Same logic. You only need to understand one thing. When there will be energy deficiency in the cell, Krebs cycle will occur. Okay, because energy deficiency is there, the body want to synthesize more ATPs. Okay, if there is excessive ATPs or sufficient ATPs in the body will try to prevent or inhibit Krebs cycle so that more excessive ATPs will not occur. This is the simple logic. Okay. Next, bioenergetics of Krebs cycle, which we have already discussed, that from one acetyl coenzyme A, three NADH are produced, one FADH2 molecule is produced, one GTP molecule is produced. One NADH molecule, when passes through electron transport chain, it will give 3 ATPs. So when 3 NADH molecule will pass through Krebs cycle, it will give 9 ATPs. When one FADH2 molecule passes through electron transport chain, it gives 2 ATPs. When one GTP molecule passes through electron transport chain, it gives 1 ATP. From one acetyl coenzyme A molecule, we are getting 12 ATPs. From 2 acetyl coenzyme A molecule, we will get 2 into 12, that is 24 ATPs. Okay. And that is what I have mentioned it over here. We are getting 24 ATPs through Krebs cycle. Okay. Other important information I have mentioned only up till now. This was asked in the previous year exam. Okay. So you may get this. You can remember if you want. You can memorize these values. You just have to memorize this one. Okay. This we will see how it is calculated. So 2870 kilojoule heat is liberated when one molecule of glucose is burned in a calorimeter. One ATP has 30.5 kilojoule energy because of the presence of high energy phosphoanhydrate bond. We will discuss phosphoanhydrate bond when we will study bonds. Okay. From one molecule of glucose oxidation, 38 ATPs are formed, which results in 1159 kilojoule energy. Okay. So 1159, if one ATP has 30.5 kilojoules energy, if 38 ATPs are formed from one molecule of glucose oxidation, we will get 1159 kilojoule. Now you understood from where this 1159 is coming from. Okay. Bioenergetics of entire glucose oxidation. We know that 2 ATPs are utilized in glycolysis. In the first step, 1 ATP. In the third step, 1 ATP. Total 2 ATPs are used. 4 ATPs and 2 NADH are produced in energy generation or payoff phase. Okay. We know that 1 NADH will result in the formation of 3 ATPs through electron transport chain, and these 4 ATPs. Okay. So if 1 NADH will give 3 ATPs, so 2 NADH molecule will result in the formation of 6 ATPs. 6 plus 4, 10. 2 ATPs are getting used. Net ATP gain is 8 ATPs in glycolysis. In glycolysis, 2 molecules of pyruvate is formed. One molecule of pyruvate, when oxidized, will result in the formation of 1 acetyl coenzyme A. 2 molecules of pyruvate, when oxidized, will result in the formation of 2 NADH molecule. 1 NADH molecule produce 3 ATPs. 2 NADH molecule will produce 6 ATPs. In Krebs cycle, we have discussed. In from one acetyl coenzyme A molecule, 3 NADH, 1 FADH2, 1 GTP is produced. But 2 acetyl coenzyme A molecules are present. So 6 NADH, 2 FADH2 and 2 GTP molecules will be produced. And we have discussed it. 24 ATPs we are getting from 2 acetyl coenzyme A molecule. Okay. So that 24 ATPs, these 6 ATPs from pyruvate oxidation, and these 8 ATPs from glycolysis. Total 38 ATPs we are getting from complete oxidation of glucose. One molecule of complete oxidation of glucose. Now, these 38 ATPs are according to old ratio. If a question is asked, calculate the number of ATPs which are formed from complete oxidation of glucose according to new ratio, then the answer should be 32 ATPs. According to old ratio, we take 1 NADH form 3 ATPs. According to new ratio, it is 1 NADH form 2.5 ATP. 1 FADH to form 1.5 ATP. 1 GTP from 1 ADP. According to old ratio, it was 3, here 2 and 1. So only if a question is specifically asking you that calculate the number of ATPs according to new ratio, then you have to calculate the number of ATPs using these values. Otherwise not. Otherwise you have to use old ratio values. Okay. So that was the last slide. I hope this was very much helpful for you. This lecture was very much helpful for you, especially for the GATE Life Science students who are taking biochemistry as their one of the subject. Okay. If you are a new student, I would advise you to watch all the videos right from the beginning so that all the concepts will be very much clear for you. Okay. And also, if you are a new student, I have one Telegram group where I regularly upload quiz, and it has more than 3000 premium quality questions. You can solve it if you want, because it will be very much helpful for you. This is my Telegram group ID or name. So what you have to do, install Telegram app. In the search button, type down this and you can join. Okay, it will be very much helpful for you. I also upload short notes, tricks and upcoming exam updates. Okay, so I hope you found this video very much helpful. Please don't forget to like, share and subscribe to my YouTube channel, and also don't forget to press the bell icon so that you won't miss any notification when my next video will get uploaded. Okay, see you in the next video. Thanks for watching.